plant tissue cultures and their uses. But first, what is plant tissue culture? Plant tissue culture is an aseptic technique in culturing plant cells, tissues, or organs on artificial medium under controlled nutritional and environmental conditions. There are several types of plant tissue culture. Let us take a look at the first one. Embryo culture. Embryo culture is an in vitro technique of growing embryos aseptically on an artificial medium. There are two types of embryo culture. One, a mature embryo culture, and the second one, immature embryo culture. Mature embryo culture is where a mature embryo derived from a ripe seed is cultured. This is to avoid inhibition in the seed for germination. As for immature embryo culture, is a culture that grows immature embryos originated from unripe hybrid seeds which results in germination. Culturing of embryos include excision of embryo and culture requirement. Excision of embryo depends on whether the embryos are mature or immature. Culture requirement is the selection of medium in order to support the progress and development of the embryos that is excised in different stages. The zygotic embryo development is as follows. First, globular, hard, torpedo, and then cotyledonary. The embryo will be cultured in the dark at 25 degrees Celsius until the seedlings have grown 2 cm long and formation of fruit starts. Once this happens, the seedling will then be cultured under the light until it can be planted into the soil. There are several applications of embryo culture, such as crop improvement, in obtaining hybrid plants, and to develop disease-resistant plants.
In plant tissue culture, explants, small part of plant tissue from leaf, root, or shoot are allowed to multiply on nutrient medium. The formation of dividing cells are called callus. Callus is known as irregular, unorganized, and undifferentiated mass of actively dividing cells. Generally, callus are produced as a result of wounding or of culturing tissues in the presence of growth regulator. Callus formation is induced from plant tissue samples explants after surface sterilization and plating onto tissue culture medium in vitro. Callus can directly undergo a direct organogenesis and or embryogenesis where the cells will form an entirely new plant. This process is known as phagogenesis, where the cells will form an entirely new plant. This process is known as callus culture.
What is cell suspension cultures? Suspension culture is a type of culture in which single cell or small aggregates of cells multiply while suspended in agitated liquid medium. It is also referred to as cell culture or cell suspension culture. The cell suspension culture requires optimization of the cell line, the cultivation media, and bioreactor system. There are two types of cell suspension cultures. First, batch culture, and second, continuous culture. Batch culture is a closed system culture. In this system, the cells are grown in a fixed amount of culture medium under suitable environmental conditions. These are used to initiate single-cell cultures. The batch culture follows an S-shaped graph. it is consists of few phases. First, lag phase. Second, log or exponential phase. Third, stationary phase. And fourth is death phase. During the lag phase, the cells adjust to the environment and prepare to divide. During the log or exponential phase, the cells divide and enlarge rapidly using the growth medium. In stationary phase, the cells stop dividing after 3 to 4 generations of cell division. And the last phase is death phase, where cells start to die. For the continuous culture, it can be open or closed system cultures. In these cultures, the fresh medium is continuously aided and the detoxification of the media is avoided by continuously removing leftover nutrients and metabolic end products from the medium. There are two types of continuous culture, which are closed system and open system. Closed system is where the addition of fresh medium is balanced by the outflow of old medium. So in this case, cell biomass continues to increase with the growth of the cell. For the open system, the balance is maintained by adding the fresh medium while harvesting the same amount of the culture, containing both cell and the medium.
Protoplast culture. The definition is protoplast refers to spherical plasmolized content of plant cell enclosed by plasma membrane without cell wall. Application of protoplast culture involve the regeneration of complete plants and production of hybrids of sexually incompatible species. Steps involved in protoplast culture is protoplast isolation, protoplast purification, protoplast fusion, and selection of somatic hybrids. In the isolation of protoplasts, it involves mechanical methods and enzymatic method. In mechanical method, first step, plant tissue immersed in 1 molar sucrose until protoplast shrunk from the enclosing. Second step, plant tissue cut with a sharp knife. Only cell walls are cut without damaging the protoplast strips. And the last step, protoplast will be released by osmotic swelling. Enzymatic method. First step, enzymes added to prevent plasma membrane from rupturing. Second step, 50 mm calcium chloride added to increase stability of released protoplast. Third step, incubate tissue to release the protoplast from the tissue. Protoplast purification involves removal of debris and enzymes and removal of broken protoplasts. Removal of debris and enzymes. First, debris are removed by filtration with nylon mesh of 100 micropore size. Second, enzymes are removed by centrifugation at 600 rpm for 5 minutes. Removal of broken protoplasts. Suspending the protoplast, preparation in 20 until 40% sucrose solution and centrifuge at 350 rpm for 3 minutes. After the purification of protoplast viability and cell wall formation test is being proceeded. Add 0.01% calco flour solution and observe the protoplast under a fluorescent microscope. The cell wall will fluoresce while the protoplast will remain dark. There is 3 ways to do the protoplast fusion. First, in a high calcium and high pH solution, in a polyethylene glycol, and the last is electric field. Selection of somatic hybrids. First step, culturing the somatic hybrids where only somatic protoplasts can grow. Second step, complementary selection on specific culture medium. Third step, mechanical isolation. Fourth step, morphology of the plant.
Root culture. Root culture can be defined as the culture of excised radical tips of aseptically germinated seeds in a liquid medium where they are induced to grow independently under controlled conditions. Root tip cultures are generally maintained in moving liquid medium. In culture, root tips are induced to grow the root system of an intact plant. A clone of excised roots can also be established from a single root culture by repeatedly cutting and transferring of the main root tips or of lateral tips into fresh medium in every subculture at the interval of definite period. Examples of plant species that can be root cultured: tomato, carrot, peas, and clover.
What is Meristem culture? Meristem culture is the vital culture of a generally shiny special dome-like structure measuring less than 0.1 mm in length. It involved the cultivation of the shoot apical meristem. This technique also refers to shoot tip or apical meristem culture. Meristem is the shoot tip or the apical region which is found at the shoot apical and root apical region. It is dome-shaped, whose thickness is about 0.1 mm in diameter and 0.25 to 0.3 mm in length. The technique of meristem culture is very much similar to the technique of micropropagation, which also involves initiation of culture, multiplication of culture, rooting of a developed shoot, and transfer of plantets to the pots.
The process of meristem culture technique involves the following steps. First, remove the young twigs from the healthy plant. Then, cut the tip portion of the twig that should be 1 cm. Then subject the shoot tip for the surface sterilization in a sodium hypochlorite solution for 10 minutes. Rinse the eggplant thoroughly with the distilled water for about 4 times. Then transfer each eggplant to a sterilized petri plate. Dissect the outer leaf of the shoot tip. After the dissection of outer leaf, the apex region will get exposed, which is separated by the help of a scalpel. Transfer the apex or apical meristem to the MS nutrient medium. Then incubate the culture under 16 hours light at 25 degrees Celsius. After the development of single or multiple shoots, transfer it to the hormone-free medium for the root development. Then, transfer the plants to the pots containing compost and kept under greenhouse conditions for hardening.
The application of shoot tip or meristem culture are virus elimination, micropropagation, storage of genetic resources, used in plant breeding, and for quarantine.
Next, we are going to talk about the microspore culture and its uses. Microspore culture, which also known as pollen culture, is a technique that separates haploid embryos by embryogenesis. This technique allows us for speeding production of completely homozygous lines while in the context of expanding biology. There are several ways on how to separate the pollen grains, either by mechanical methods, split methods, or flood culture methods. Flood culture method is way better than mechanical method because it doesn't damage the pollen grains. Microspore culture can be used in protoplast isolation, transformation of transgenic plants, or as crop improvement in vegetables and cereals. The benefits of microspore culture are less competition among the microspore, no diploid anther walls, and better potential haploid plant production.
For the conclusion, there are many types of cell cultures, and each of these cell cultures has its own unique functions. Tissue Tissue culture is therefore of great significance in biological studies due to its wide range of applications. Most importantly, tissue culture produces a pest and disease-free seedlings. Thank you for watching.
왠지 쌉쌀해 근데 또 달콤해 내가 지금 다크 초콜릿을 먹고 있는지 곁에 있으면 녹잖아 말 한마디에 흐르잖아 난 네가